Genetic variants are found all over the genome. For most variants, it is difficult to know the consequence of small changes in a DNA sequence, because the function of many sequences is unknown. This is not the case for exonic variants in genes, as we can anticipate what the consequences of changes in the coding DNA sequence are for the polypeptide that will be made. We distinguish three types of changes in the DNA sequence on the nucleotide level: insertions, deletions, and substitutions. As the names suggest, insertions are variants that have additional nucleotides inserted into the reference sequence, whereas deletions differ from the reference sequence because nucleotides are missing. Insertions and deletions, or indels, cause a change in the length of the DNA sequence. When this happens in sets of 3, the reading frame is not affected, and the following sequence will be translated normally. Such indels are in frame. However, when the reading frame is affected, the translation of the subsequent codons is changed. The resulting protein will then be drastically different. This is called a frame shift. Substitutions are variants that only have one nucleotide changed compared to the reference sequence. Depending on the position in the codon, the substitution of a nucleotide may cause the change of one amino acid in the translated protein, which is known as a missense variant. It may also cause no change, as multiple codons can encode the same amino acid. This is known as a silent variant. In some cases, a substitution creates a premature stop codon, which causes the termination of translation, leading to a shorter protein to be formed. These variants are called nonsense variants. Frame shift variants may also create a premature stop codon further downstream of the indel. When a premature stop codon arises at the beginning of a gene, the whole mRNA strand may be discarded through a mechanism called nonsense-mediated decay. This variant then forms no protein at all. Since different exonic variants have different consequences for the translated protein, exonic variants can both be harmless or harmful. This outcome depends on different factors, such as the type and location of the variant. You can probably imagine that nonsense or frame shift variants are generally more harmful than silent variants, and that a missense variant in an important part of the protein is more harmful than one in a less important part. When talking about genetic diseases, note that a certain gene is associated to a disease. This does not mean that each variant that is found in that gene causes disease. To determine whether a variant is disease-causing, we classify them in 5 categories, ranging from benign to pathogenic. When a variant is classified as pathogenic, there is enough evidence to conclude that the variant severely disrupts the function of the protein and thereby causes a Mendelian disorder. Pathogenic variants, and thus Mendelian disorders, are on a population level very rare due to the large molecular impact that they have. Remember this graph? Pathogenic variants are often located in this corner of the graph. As the name implies, benign variants are not causing Mendelian diseases. So, to conclude, exonic variants have a wide range of effects on protein level, which can be partly understood following the central dogma. A variant can disrupt the function of the protein severely, causing a Mendelian disorder.